let's dive into a really fascinating puzzle right at the heart of chemistry. We're going to explore how things can be made of the exact same ingredients, but end up being completely different. It's kind of like a molecular identity crisis.
So think about this for a second. How is it even possible that two totally different sugars, which our bodies use in very different ways, can be made of the exact same atoms? I mean, on the surface, that sounds impossible, right? Well, to get to the bottom of this, we're going to look at a classic example, glucose and fructose. You've definitely heard of both of them. They are fundamental sugars for sure, but they are not the same thing. And yet, on a chemical level, they kinda are. Check this out. C6H12O6. That's 6 carbon atoms, 12 hydrogen, and 6 oxygen. This one little formula can build glucose, the sugar that powers our cells. But that exact same set of parts can also build fructose, the sugar you find in fruit. Same parts, totally different products. So what's the secret? Well, the answer to this entire mystery, it comes down to a single, really powerful concept in chemistry: isomers. This is the key that unlocks how nature creates so much incredible diversity from, a pretty limited set of atomic building blocks. So here it is, the formal definition. Isomers are just compounds that share the exact same molecular formula, but have different structural forms. It's not about what they're made of, it's all about how they're put together. That's the crucial part.
Okay, so let's make this crystal clear. Let's go back to our two sugar twins. The specific type of isomerism that separates glucose from fructose has a name. It's called aldose-ketose isomerism. Let's actually see what that means.
So on one side, we've got glucose. It's known as an aldose. Why? Because it contains something called an aldehyde group. Then on the other side, you have fructose. It's a ketose because it has a ketone group. These two little groups, that's where all the magic happens.
Now take a look at this image. This is where it all clicks into place. See the glucose molecule on the left? That aldehyde group is sitting right at the very top. Now look at the fructose on the right. See that ketone group? It's just one carbon down from the top. That's it. That is the entire difference between the two. So it all boils down to this. The only structural difference between these two sugars is the position of one single double-bonded oxygen atom. In glucose, it's on the end carbon. In fructose, it's on the second carbon in the chain. It is a tiny, tiny shift, but with absolutely massive consequences. But wait, it actually gets even more interesting. Aldose-ketose isomerism is just one type. There's another fascinating layer to how molecules can be the same, but different. The rabbit hole goes a little deeper. And that next layer is called stereoisomerism. Now in this case, the atoms are connected in the exact same order, but they're arranged differently in 3D space. The best way to think about it is like your left and right hands. They're mirror images, but you can't perfectly stack them on top of each other. And the key to this is a special little thing called an asymmetric carbon atom. And another key term you'll hear for this is chiral carbon, which is really just another name for that asymmetric carbon we just mentioned. It's basically a carbon atom that's attached to 4 different groups, and that's what allows for these cool mirror image arrangements. It's a whole other level of structural difference.
So what's the big picture here? Why does any of this matter so much? Well, it all comes down to a fundamental, absolutely crucial principle in both biology and chemistry. So let's recap the key takeaways. First, we know isomers share the same atoms, but they have different 3D shapes. Second, and we saw this so clearly with glucose and fructose, a tiny little change in that structure can create a completely new substance with totally different properties. And this leads us to the most important point of all. In the molecular world, arrangement defines function. Structure is everything. It's why one molecule tastes sweet and its isomer doesn't, or why one drug works and its mirror image is totally useless.
And that leaves us with a final thought to chew on. We've seen how just a subtle little atomic shuffle can completely change a molecule's entire identity. It really makes you wonder, doesn't it? What other huge differences in the world around us boil down to a simple, elegant twist in structure, just hiding there in plain sight?